Okay, welcome back everyone. So now we will continue with the topic cell division part 2, which in last video we actually talked about the interphase, which is before our M phase. So M phase, which is what we are going to talk about today, is mitosis. All right. So first, let's talk about the outline of this video. So at first, we are going to talk about what is mitosis. And next, we're going to talk about what are the differences between mitosis and also cytokinesis in animal cells and also plant cells. Do you still remember what is cytokinesis? Cytokinesis happens after mitosis, which is after the cell division. Cytokinesis is the divisions of the cytoplasm. And mitosis involved the divisions of the nucleus. So what is mitosis? Mitosis is defined as a divisions of the nucleus. So nucleus is the important keywords here of the parent cell into two nuclei. So this is an overview of mitosis. So the interface, which we already mentioned in the last video, you may check it out. An interface is where the chromosomes in the nucleus, they gain energy and then they undergo three phases: G1, S phase, and also G2 phase. And after that, they're going to enter the mitosis phase. And most of the cell cycle, most of the time, takes place in the interphase. And next, they're going to progress into prophase, and then a metaphase, anaphase, and also telophase, and then cytokinesis. So mitosis basically covers four phases, which we'll talk in details later, which is the prophase, metaphase, anaphase, and also telophase. I always remember this as PMAT. Okay. So next, this is the prophase. So prophase, what happened in prophase is that the nucleus, where the chromatin starts to shorten and thicken to form chromosomes. Okay. So what is chromosomes in this prophase? So we know that the genetic materials inside the nucleus, we call it as chromosome, but in prophase, the chromosomes is we're going to call it as sister chromatids. It's called sister chromatids because they look like this, a set with um, kind of like two pairs, but they are actually one pair. So these are sister chromatids in the whole round thing here is a nucleus, all right? And then the sister chromatids, they join together at the centromeres. Can you see that there's a center, like white round shape things here in the middle that join the sister chromatids. These are what we call as a centromeres. And then the nucleus membrane and also the nucleolus, they're going to disintegrate. So the components for nucleus, we have a membrane for the nucleus, and also we have a nucleolus, which is inside the nucleus. So both of these, at the phase of prophase, they're going to disintegrate, or in another word, they're going to disappear. And then another important cell component in animal cells is centrioles. So the centrioles, they will go to the opposite side, okay? And then they will form spindle fibers or spindle apparatus, which is essential for our cell division. Do you remember that centrioles only happens in only present in our animal cells, where the plant cell, they will not have any centrioles, okay? This is an important features for animal cell. So important thing is that central is going to move to the opposite pole, which is either the lower pole here or the north pole here, all right? So, and then the chromosome going to exist as sister chromatids. So this is what going to happen since in prophase. And then next, we're going to move on to metaphase. So what is the important keywords here in metaphase? Just remember the words line up. Okay, you can see this is the photo, picture for metaphase, where the chromosomes or the sister chromatids, they are lined up, or in another word, we call it as a line. They becomes a line in a single row on the equatorial plane. You can see that all the plane here, all the lines here, they are the equatorial plane, which is formed by the spindle fibers. And what forms the spindle fibers is our centrioles. You can see the red colored things here at the two pole. They are the centrioles, and then they form the spindle fibers, which provides a plane for our chromosomes or our sister chromatids to line up. Okay, so the important words is line up or aligned in metaphase. And then the metaphase is going to end when the centromeres begins to divide. Later in the next phase, which is anaphase, the sister chromatids is going to divide at the centromeres. Okay, I will show you later. But remember that metaphase, the second phase, important word is align. Okay, next, moving on to anaphase. The centromere is going to divide into two, and then the sister chromatids, they're going to separate. Remember just now they look like an X shape. Now they separate at the centromeres at the center, so they becomes like just one string or one thread here. So this is still chromosome, but they are no longer sister chromatids. So they separate at the centromeres, and then the spindle fiber is going to shorten. You can see some of the spindle fiber here, they shorten, and then the sister chromatids, they are attracted, or the chromosomes, they are attracted to the opposite pole, okay? So they will attract to the opposite pole or to the directions of the centrioles, okay, when the spindle fiber shortens. And lastly, the anaphase going to ends when the chromatid arrive at pole. So when all these chromatids or chromosomes, they reach the pole, both of these pole, this is where anaphase is going to progress into telophase, right? And next, we're going to talk about our last phase in our M phase, or the mitosis, is that the chromatids now, we no longer call them sister chromatids. We call them as a daughter, okay? Just, do you understand? Daughter chromosomes, okay? Because they are single straight. If there are two threads here, we call them as a sister chromatid, okay? But now we call them as a daughter chromosome. So the chromosome going to reshape again and form as a chromatid threads, okay? Where a chromatin is a chromosome that can be seen, that is visible under a microscope, okay? That's what, by the meaning of chromatin. So the nucleoli and also nuclear membrane, remember that at the prophase, they all disintegrate. So now they kind of form again, and then the spindle fibers going to disappear. You see, there's no spindle fibers, and the nuclear membrane, the nucleolus, they're going to start to form back. And then telophase ends and going to continue by cytokinesis. You can see it now, we have two nucleus, but they are still kind of connected because the cytoplasm is not yet separated. So after this telophase, or after the whole mitosis, we're going to continue with cytokinesis, where the cytoplasm going to separate, and then they're going to form into two separated nuclei. All right. So next, we're going to talk about cytokinesis, and we're going to uh, learn on how to differentiate mitosis and cytokinesis in animal cells and also plant cells. So remember that only animal cells have centrioles and plant cells do not have centrioles. So how are they going to undergo this cell division? Although they do not have centrioles, they still can form spindle fibers during mitosis, and everything is still uh, is still the same in cell division for plant cells, all right? But just remember that they do not have centrioles, yeah? Okay, so moving on to cytokinesis. Cytokinesis, which is the separation of the cell division, they are different in animal cells and also plant cells. So we talk about animal cell first. So divisions of cytoplasm after telophase, good, cytokinesis. So plasma membrane going to constrict in the middle. So constrict in the middle is the key words here. You can see in the middle, which is the blue, light blue color here is the cytoplasm. So the plasma membrane here going to constrict in the middle, okay? Going to constrict where we have a contractile ring creating a cleavage furrow. A cleavage furrow is that they're going to becoming narrower and narrower, and then they're going to cleave, or they're going to separate. Okay, so they're going to constrict, and then they're going to slowly separate, and then there are microfilaments in the center. So the microfilaments going to contract as well, which cause the cell to constrict, and then lastly they're going to split and forms two daughter cells. Okay, so you have the contractile rings here, and you have a microfilaments contracts, and then they're going to cause the cell to constrict until they becomes a two separate daughter cell. And next, talking about cytokinesis in plant cell. See, we still form two nucleus after mitosis, but the cytoplasm is still connected here. So the vesicles going to combine to form the cell plates. So the important keywords here is cell plates. So you can see all these little dots here, they are vesicle, but then they're going to combine together and form a cell plate. Okay, becomes a plate here after the vesicles combine. So the cell plates are surrounded by new plasma membrane, and then they form a new cell wall. And so they're going to become two separate cells, okay? But you still remember plant cells are having plasma membrane as inner layer and also a cell wall as an outer layer. So the cell plates, they're going to expand outwards. You can see they are still not connecting to the border. So these cell plates, after fusions of the vesicle, they're going to expand outwards until they combine with the plasma membrane. And thus, after this, they have their own cell wall and own plasma membrane for two separate cells. And after reaching the border, they're going to form two daughter cells. Okay, so important keywords for plant cells is the cell plates, and important word for animal cells is the microfilaments contract in the middle. Okay, and both going to form two separate daughter cells. Okay, that's what common in features for animal cells and also plant cells. So summary of this video, what we learned. First, we learn about M phase. Remember the mnemonic, which is the PMAT, prophase, metaphase, anaphase, and also telophase. That's the overall for the M phase, mitosis. And then next, we talk about the differences between mitosis in animal cells and plant cells, which they are both actually the same. They still form spindle fiber, but just that plant cells do not have centrals, and animal cells have centrals that go into migrates to the pole, okay? And then the chromosome is going to be attracted to the poles as the phases progress. And lastly, for the last lesson, we learn about the differences between cytokinesis of animal cells and plant cells. So cytokinesis, the separation of cytoplasm. Animal cells, they are microfilaments that go into contracts. There are contractile rings that go into constricts in the middle and form two cells. While for plant cells, there's vesicles that go into combine. Vesicles, the little small dots in the middle, they combine and form a cell plate. And then the cell plate is going to fuse to the border. They go into contact with the plasma membrane, and the two cells going to develop their own cell wall and also own plasma membrane. So the end result for both animal cells and plant cells after cell division, after cytokinesis, is going to be two daughter cells, correct? Just remember that the outcomes for mitosis is two daughter cells. So that's all for today's video. I hope you learned something. Thank you very much. See you. Bye bye.